Perfect. Thank you very much. So I'm going to talk to you now about what we're doing here at Evox Therapeutics. So what we're really focused on from a company perspective is developing a new class of drug around something called an exosome. And so I'll talk to you first about what they are and how we think they can be transformative. So exosomes are these small nano-sized vesicles, typically 30 to several hundred nanometers in size. And they are small vesicles containing a membrane. They contain proteins and nucleic acids, and they're naturally secreted by all cells. And as they get secreted by cells, they also get taken up by cells. And it really represents the way the body and cells talk to each other. And so as a pathway, exosomes are conserved across species. They're even conserved down into plants and fruit. So wherever you've got an organism with more than one cell, this is the way nature has really allowed cells to talk to each other. What we're doing is we're using this natural, it's very efficient and very safe pathway to deliver drugs. And those drugs can be proteins, they can be nucleic acids. As you'll see, we've actually adapted it to be able to deliver practically any type of drug. And so in this way, we're now able to make drugs where we can deliver drugs to places they normally don't get to. So if you think of proteins, biologics, they're typically not good at crossing the blood-brain barrier. We can do that. Or we can target nucleic acid drugs like siRNA, mRNA outside of, for instance, the liver, which is where the technologies only evolved to get to that one organ. We're able now to get these to a wide variety of organs, and we're also able to target them so they can preferentially go to particular cell types or to specific tissues. And so we're really focused from an Evox perspective on delivering biologics and nucleic acids, typically for our own pipeline, looking at proteins and messenger RNA molecules. With this type of platform, we think we can go extremely rapidly. We think we can start programs and get them into man in 18 to 24 months. Because, again, as you'll see, it's a very modular platform. So as we understand how it works, to make a new drug will literally take us a few months. We're focusing on the rare disease space for a lot of obvious reasons. The first and foremost is we know exactly for most of these genetic diseases, we know what the drug is or needs to be. We can make that in the span of a few days. And it also means that from a risk perspective, there's no what I call biology target risk. It all becomes a fundamental pharmacology risk, is can the exosome deliver the drug to the right cell at the right time? And that's something you can very easily follow preclinically. And so that's the only risk. Obviously, longer term ambition is to take that proprietary pipeline and advance it ourselves and ultimately commercialize it. And so that's something then by focusing on the rare disease space, we're able to potentially move all of those programs forward. We've been doing this, as you'll see on the next slide, for about 10 years, not as Evox, but if you go back to when the founders first started. As a consequence, we built up world-leading expertise in how to engineer these exosomes to contain drugs. And with that comes really broad, dominating IP. And obviously from a basically management team perspective, we've done this, and I've done this multiple times with new platforms like siRNA, mRNA, single domain antibodies. And so, we really see this as potentially being just as transformative. This is the history of Evox itself. The original scientific discoveries date back to 2010, but Evox itself was founded in 2016. We closed a large Series B, September of 18. And as you'll see, we're really planning a whole slew of pipeline INDs and CTAs over from the end of 2020, 2021, and basically beyond. This is really what I've talked about here in terms of the focus on our proprietary pipeline. And beyond that, obviously, this sort of platform can be used to kind of deliver all sorts of drugs. If it's an siRNA drug, a small molecule, it can also be used to treat broad CNS kind of diseases, oncology, a whole slew of things. And so that's where the partnership model comes in, where obviously there are a lot of pharma, large kind of biotechs who have drugs that they've made, that look quite good, but what's holding them back is the inability to get them to the right place, and that's where we can really help move that forward and use that platform and those sorts of partnerships to just to catalyze our own growth, but also to use that as an opportunity to learn more about what the technology can and can't do. This gives you a sense as to how we actually make and load drugs into these exosomes. So basically, as I mentioned before, all cells produce exosomes. So our cell source of choice is really going to be a kind of human cell line, allogeneic line. So we're not taking cells from patients. We're just taking a cell line, the same way that you take a cell line to make an antibody. And in this case, we engineer that cell to make the drug that we want expressed. And the real trick here is that we've understood what signals need to be incorporated into that drug to have the cell incorporate it and load it into the exosome that it's naturally producing. So it's basically making what's called a stable cell line, where the cell's making the drug in a format where the cell makes the drug, makes the exosome, loads the drug in the exosome, secretes it, and all we have to do is purify the culture supernatant, and we get our drug at the end of the day. What's nice about that from a process perspective is that the front half is all very simple. The back half, in terms of how you actually purify the exosome, doesn't really change from drug to drug. And so from that perspective, once you've worked that out, you can just iterate very rapidly. To give you a sense as to the sorts of things and the amounts we can load, we're really talking hundreds of thousands of copies of these drug classes per exosome in a very homogeneous manner. What are the sorts of things that we've been able to do? And most of this we've already shown from a pre-clinical in vivo perspective, the ability to get proteins across the blood-brain barrier after you give them systemically. We can load these exosomes with hundreds of different copies of the same drug, or we can decide we want to load 2, 3, 4, 5 drugs. Basically, there's no kind of limit as to how much you can engineer in. So you can make multivalent, multi-specific